Welcome everybody. My name is Eriko Takano and I am the coordinator for the TOPCAPI project, the thoroughly optimized production chassis for advanced pharmaceutical ingredients. And I will share with you a slide just to introduce you to the webinar. So I hope you can see the slide. Yes. So welcome everyone, and thank you for coming for this Microbial Cell Factories webinar series. So this is organized by the Horizon 2020 Industrial Biotechnology Project. So myself and also Professor John Morrissey, who is the coordinator for CHASSY project, which is the model-based construction and optimization of versatile chassis yeast strains for production of valuable lipid and aromatic compounds. I just want to give you a quick introduction about TOPCAPI, and John will be giving an introduction about CHASSY in the next webinar date. So TOPCAPI is with 8 different partners from 8 different countries, sorry about that, with universities, academic institutions, and also 3 small and medium enterprise. So our really aim is to use systems biology and synthetic biology using this design, build, test, and learn cycle to produce an antibiotic high production chassis in Streptomyces. And our target is industry and related because the compounds that we're trying to isolate, sorry about this, is the targets that the industry are interested in. And this brings me, this design, build, test, learn cycle, what we're using brings me to our webinar. So our aims of our webinar is to have a microbial cell factory development for concept to commercial development and all stages in between. And so we would like to have experts from academia and industry to share their knowledge on the 3 different themes. So today's theme is going to be on in silico design and reprogramming of microbial cells. The next webinar, the 25th of March, will be on strain engineering approaches to build cell factories. And the last but not least, webinar is on the compound analysis and transferring technology to commercial scale, which is on the 8th of April. So I would like to, this is our schedule for today. So the first speaker is Professor Jean-Loup Faulon, and followed by Professor Rainer Breitling, who is a consortium partner for TOPCAPI project. And last but not least, by Yvan Domenzang from the CHASSY project. And we can close with either general discussion and a close of the webinar. So just a few things to let you know. First of all, that though you might not be able to see everyone, all the participants that's here, we had about a registration number of people of about 140. So there's more than 100 of you, I hope, who is listening to this webinar. Another point is that this webinar will be recorded, and we hope to upload this onto our TOPCAPI and CHASSY websites so that you can follow it again. And also, after each question, there will be time for a question to ask questions. But we want you to put your questions into the question and answer box. You'll be able to find this in your, I don't know where, right hand, bottom, top, but not in the chat box. There you won't be able to find the chat box, but please put your questions in the question and answer box. And I will read them out to you, and hopefully the speakers will be able to answer them. Okay, so that's all from me about the general idea of the webinar and to welcome you. And so first, I'd like to introduce to you the first speaker, our keynote speaker, who is Professor Jean-Loup Faulon. So Jean-Loup is a senior research director at the French National Institute. I will stop sharing my thing so that Jean-Loup can share his slides. So Jean-Loup is a senior research director at the French National Institute for Agronomical Research, the INRA. And he's very well known for his work on computer-aided design and retrosynthesis in biology. And today, Jean-Loup is going to talk to us about automation workflows for synthetic biology and metabolic engineering, the Galaxy-SynBioCAD portal. So Jean-Loup, please.
So thank you, and thank you, Enrico, for this nice introduction, and also to give me the opportunity to present some of the recent work we have been carrying out here in INRA. So since it's a session about design, in silico design, so I thought it would be appropriate to talk to you about a system we recently launched. So it's available and free of use, which basically compile various softwares that have been developed throughout the years, and that have been, and that's the difference here, that have been encapsulated into a Galaxy system. So if you are interested further by this, you have here a web address, and you can go there and start to use the system. It's free of use. You just have to register, but you can freely use the system that I will present. So so it's been some time, that people have developed many software tools in the context of synthetic biology, and in particular metabolic engineering. I will focus more on that than the other aspect of SynBio. And so there are plenty of tools, okay? And but there is no really even a list, or even reviews, exhaustive reviews of all the tools that exist. So this is just my take on the subject, And so these tools can, of course, deal with the design aspect of the DBTL cycle, but also some of the tools, are drivers to robots, or can automatically build protocols, which are more linked to the build stage. And some other tools can also be used to analyze the data that is produced during the test part of the cycle. So there are many tools, okay? So they are very much scattered. Sometimes they are redundant, so it's difficult to decide which one to use. And another difficulty is that some of the tools require some expertise actually to be run, as far as the input is concerned, what they generate, and so you basically need help from bioinformaticians to run these tools. Some even require some computing resources, and you need computing power to use these tools. But what's most important is that these tools, although they seem to cover many aspects of the DBTL cycle, they are not interoperable. That is, the output of a given tool will not necessarily go into the input of the next tool, even though they seem to be appropriate to link them together. So when we look at all these tools, we also put some constraint because we wanted to plug them into the Galaxy system, and I'll explain later what it is. But basically, we wanted them to be open source and to be under MIT license, so there is no restrictions on the usage of the tool or on the modifications of the tool, because we actually needed to modify some of these tools in order to standardize their input-output, in order for them to start to communicate between each other. And also, we wanted the tools to be well published, validated, and have somehow a user community. So that basically limited the number of tools that could be that could fit all these categories, and these are those that are listed now on this simplified slide. So what I'm going to go now is to go through these various tools, and in particular those that we have already implemented into Galaxy, and tell you a little bit of what they do. And there are roughly three categories of tools. The first one are dealing with finding solutions to synthesize a molecule into a given organism, provided that you are plugging in heterologous enzymes. So these tools are basically belong to the retrosynthesis area of research. And there is nowadays about a dozen of groups around the world, that are developing retrosynthesis algorithm. One was recently published by by someone in Manchester very recently. And so anyway, so once you have done this, you basically get all the possible pathways that will link your chassis organism to the target molecule you wish to produce. And the linking is done through, of course, metabolic reactions. So then the next step is that since you have several solutions, is to analyze these various pathways. In this case, there are 11 different solutions, and some criteria might be taken into account. For instance, the enzyme availability. For some reactions, there might be no enzymes that are available to catalyze the reactions. So you need to search these. And there are tools like ECBlast, done by the Thornton group at EBI, and also Cell Enzyme down in Manchester by Pablo Carbonell that can do the job. So what these tools will do, they will give you back a rank list of enzyme catalyzing the various steps into your retrosynthetic map or into your pathway. There's also some other criteria like the thermodynamic feasibility of the reactions. So there are tools like Equilibrator done at the Weizmann Institute that can actually compute for you the free energy of the various reactions. And finally, different pathways can yield to different, can produce different yield, and that can be analyzed using flux balance analysis. And I'm sure you are all aware of the CobraPi toolbox, which allow you to look at that and to predict theoretical fluxes for the various pathways. So once you basically have done this, you can get down to a list of enzymes corresponding, say, to the best pathways. So the next series of tools will deal with the actual genetic constructions. And to start with, if you have the enzyme and you wish, for instance, the enzyme, if you are dealing with a bacterial strain, and you wish to have different level of expression of the enzyme, you can play around with the RBS. So you can plug in an RBS strength, and there are tools like Pargini, also done in Manchester, that can compute for you the RBS sequences corresponding to the strength. Once you have done this, you can also connect to SynBioHub, and still looking at the level of expression of the various enzymes, you can choose different vector, different plasmid with a different plasmid copy number. You can also select libraries of promoters of different strength, and you basically have a combinatorial space of possible constructions. And tools like DOE, also done partly in Manchester, but the work has been continuing in Spain, can actually sample the combinatorial space and produce for you a sample of construct that correspond to the space of possibilities. And these are actually produced in SBOL format. So at this stage, you really know what are the constructs according to your specifications. And you might want to choose now a protocol, an assembly protocol, to actually assemble these different constructs. So then there are a series of tools here again that can do this for different assembly protocol, LCR, LCR GNIT. This is done, it's a new software done in Liverpool, and DNA Weaver, a software that was done in Edinburgh, which can actually, depending on, which are actually taking Golden Gate and Gibson assembly. And what these tools are doing is that they are basically giving you the different pieces you need to get synthesized in order to assemble the plasmid according to the protocol that you have chosen. So then there is even tools that can take these different parts and that can actually write scripts to directly drive robots. And DNABot is a tool that has been developed at the Imperial College, which work with OpenTrons and actually can do the assembly process and even the strain transformations. And so you basically are at a stage where you can enter the system, you draw your molecule, you select the chassis, you select an assembly protocol, and it will spit out a library of different construct and even the script to actually synthesize this library. So this has been a community effort, and we have chosen to actually plug these tools into the Galaxy framework. The main reason is that it's quite easy to use. In Galaxy, you have a list of nodes that are called nodes. These are the different tools, and you just need to chain these nodes together in order to make a workflow. And what you see here is an example of a workflow that performs genetic design. So the reason we selected Galaxy as a platform is first, there's quite a large community. In Galaxy is very popular in bioinformatics, was developed for genomics, genome analysis. But nowadays it has many other applications, and some of these are quite relevant for metabolic engineering and biotechnology in general. For instance, metabolomics, metagenomics, proteomics, system biology also, and even tools for flow cytometry analysis. And another reason is that Galaxy has been chosen as the workflow system by the Elixir infrastructure. It's a big European infrastructure. And there is, of course, many people developing Galaxy tools, and this will only increase. One also mentioned that there is a community within Elixir that is dedicated to microbial biotechnology, where we are actually looking, I'm part of this community, we are actually looking at writing workflows using Galaxy, but also some other systems within Elixir. And finally, the last reason is when we released the Galaxy platform, it was quite well received by the community, by the metabolic engineering community when it was released. So now if you enter the system, once you register, you have videos that can explain to you how to run the workflows. There are 3 workflows that are available, one for retrosynthesis, one for ranking and analyzing the pathway, and one for genetic design. And you can even, there are videos to help you to build the pathways, to build, the workflows. So we also have visualization tools. You can visualize the retrosynthetic map. You can focus on the map, and you can click on the various enzymes or the various compounds. For instance, clicking on the reactions, you get a ranked list of enzymes that is spit out by the cell enzyme node. You can also visualize the SBOL construct using VisBol. It's a software that is developed at Newcastle University. And so we actually benchmark this workflow, at least the retrosynthesis and pathway ranker workflow. And to do this, we compile 100 different literature pathway, and we run the workflow on the same compound and strain, and generated about 8,000 pathway. And then we rank the pathway using the length, the enzyme availability score, the target production flux, and the pathway thermodynamics. And we found out that in the top 10 pathway written by the system, 65% were the actual literature pathway. And when we rise that up to 50 top pathway, 88% were the one found in the literature. But of course, if the system spit out a pathway that is not in literature, doesn't mean it's a bad pathway, because it just has not been done already. So for this, we recruited about 40 experts, and we asked them to validate batches of 5 pathways, and it was a double-blind test. So we didn't, we generated these randomly. We didn't know if the literature pathway was in it, and we didn't know, and of course the user, the expert user, didn't know that as well. And so once we retrieved the data, we trained a classifier, and we found actually an 85% cross-validation accuracy, which is a quite good number as far as cross-validation accuracy are concerned. So where we are now these days is that we are moving these tools into Galaxy Europe. So they are servers and Galaxy instances that have quite large computing power, and so for that reason, we decided to move all the tools which were first hosted on a cluster to the Galaxy Europe. So that necessitated to go from the Docker technology to Conda. So it took some time to do this, but that will allow us at the end to use all the Galaxy tools that are readily available. And for instance, all the tools that have been developed for mass spec analysis, for instance, and so it could be used to annotate spectra that are coming out of the test part of the DBTL cycle. So of course, we already have a system that can drive a script to drive robot, but it's limited to OpenTron. So what we want to do now in the future is to actually have a driver for other type of robots. But rather than writing a driver for each vendor, we preferred to use a standard communication language, which is called Scila. And so rather than writing directly in the language of the vendor, we write into Scila. And the good thing is that equipment vendors have already written interface between their equipment and Scila. And this is the case, for instance, for Hamilton or Tecan, you know, and there's about 67 available drivers that are already available. Also, the next things we want to do is to plug into the system an active learning. So this is the L part of the DBTL cycle in order to optimize a given process. And what I'm going to show you briefly is an example of that we have done in cell-free system, where we have looked at maximizing the productivity, simply protein production, to maximizing the productivity of cell-free systems. So I'm not going to go into the detail of cell-free, but you basically have to prepare a crude extract, and you have to add some components, some, let's say, magic components into the system in order for the system to work. That is, to work mean that if you throw a piece of DNA that is encoding for something, it will be transcribed and translated. So if what you have encoded is an enzyme, so the enzyme will be active, and you'll be able to transform a substrate into a product. And this has been done in the past, for instance, by Michael Jewett. So, but the problem is to find the right component to add into your cell extract in order for the system to work. So there are protocols out there, and here's the protocol that is given for E. coli TXTL cell-free systems. And not only give the list, but it also gives the concentrations of the various components. So what we wanted to do, and through an active learning process, was to search if there was not different concentrations of this component that would further increase the productivity of the system. And to measure productivity, we just express a fluorescent protein. And we also wanted to search if we could not find a system that would cost less than the system that is given in the protocol. And in order to reduce the cost, we decided to reduce the concentrations by 2, by 3, and by 10. And so that generated a large combinatorial space of possible concentrations, about 4 million of these. And active learning was perfectly suited in order to search that space. So how it works, so we started by basically drawing randomly about 100 different concentrations for the various components. And then it was fully automated. So we used a robot to dispatch the concentration on 384 well plate. We actually got the concentration in triplicates. And then since we expressed a fluorescent protein, we needed to incubate, and we read the fluorescence through a plate reader. So since we had now the fluorescence corresponding to the various concentrations, we were able to build a model that can predict from the concentration what could be the fluorescence. And then we use the active learning process, which makes use of the model in 2 different modes. The first mode is exploitation. So that is, we're going to use the model, and we're going to predict what is the fluorescence of the concentrations we have not tested already. And we're going to keep those that have the highest fluorescence, predicted fluorescence. And we will also use the active learning in exploration mode, where, in this case, we don't necessarily choose concentration that will lead to very high fluorescence, but we choose concentration where the prediction is not that good. So when we have a high standard deviation. And that is happening where we are sampling the concentration in part of the space we have not looked at before, and this can be captured by the standard deviation. So anyway, this is called the upper bound confidence bound, and so we can use that formula and pick the 100 best according to the UCB formula, and we can iterate the process. So what you can see here is that it takes 1 day to run because you basically need to incubate overnight. So we run the process, the fully automated process, for 10 days. And at day 7, we actually got a productivity that was substantially higher than the one of the protocol. It was actually 34 times higher than the one of the protocol. So it is indeed a good system if you want to express proteins into cell-free. And so not only we got higher yield, but as we went on, you can see that the correlation coefficient between the predicted fluorescence and the actual measured fluorescence also increased. And it went up above 0.95, which is also a very good number, considering that you are in cross-validation. This was done in 5-fold cross-validation. And at the end, we found that some of the components like tRNA, CoA, and NAD were not that necessary to be plugged in. And in fact, probably there was enough in the cell extract to begin with, so there was no need to add these various components. So this was one example of a fully automated, let's say, DBTL cycle, of course, on a simpler system than a strain engineering, but yet it was still fully automated. So I'm gonna stop here. I don't know if I'm okay on time. I have not keep track of my time, but I wanted to thank the people that work. So especially those that are on the top. They are the one that actually developed the Galaxy platform. Neil Swanson, Valentin Zulkover, and Jeff Baldwin also contributed pieces of software that are in the system. And those that are on the bottom actually either tested the system or developed pieces of software that have been plugged in. And I also wanted to thank the funding agencies, and in particular the ANR, which heavily funded this project, along with some little less money, but some H2020 project. BISBA, an infrastructure, and BioRobust is a community action program. So thank you, and I am ready to your questions.
Okay, thank you. Thank you very much, Jean-Loup. So there's a question here, which I will read out to you. Are there plans for the Galaxy platform to connect with registry platforms, such as ICE, for managing DNA parts and plasmids?
So right now we are connected with SynBioHub, okay, but we have not yet connected with the ICE is JBay, right? Yeah, we have not yet connected with JBay, but we are in contact anyway with the people, the biofoundries, that are over there in Emeryville. So that is something, yeah, we might actually do in the future. But these things take time, okay? So I already got 3 people working full-time on this, because it's not that easy to encapsulate these things into Galaxy and have a streamlined process. It's very easy to use, not that easy to develop. So we will, of course, and anyone actually that want to contribute is welcome, okay? Because this is being published in the toolshed, okay, in the Galaxy toolshed, and everybody can contribute to the toolshed, So we can take care about the connectivity, the interoperability. But if someone has a nice tool, and think it could be a valuable addition to the Galaxy platform, we are always welcome to incorporate that tool,
Okay, great. Thanks very much. We have time for maybe one more short question, if there's any questions from the audience.
Enrico, this is John. Yeah, may I?
Yes.
I think I'm not able to type a question because I'm an organizer. Okay. But Jean-Loup, this might be kind of a naive question now, but I mean, the Galaxy platform, the pipeline you have, has different softwares that you've integrated into the pipeline. And I think my question is, very often the developers of particular softwares, are continually updating them and upgrading them. And so is that incorporated into what you've got?
Yes, so we keep we keep track of the new versions of the softwares, and this is why I have dedicated people, Inra gave me dedicated people to actually run the platform, you know. So these are permanent staff engineer, you know, that are keeping track of the new versions of the software as they come in, you know. And but not all of them, you know, are like that, you know. But there's some, you know, like Cobra Pi, for instance. It's changing, you know. So we keep track of that. Yeah.
Okay, but you pretty much have to do it manually. It's not automatically getting the new versions.
We'll see that in the we have now an automated pipeline with Conda, you know. So if the software is well developed, which is not necessarily the case of everything we have in, you know.
Yeah.
That should be we should be able to automate that, but not necessarily for everything, you know. Okay. And also some detail that I did not go into, you know, but there was a lot of work done in order to standardize the input-output, you know. So we created specific format in the annotation of the SBML, you know, in order to take care of all the information, the enzyme sequences, you know. This is not normally what you find in SBML file, but we had to do all of this. And we also had to write a converter between SBML and SBOL, you know. So these things are not that, they seem easy like that, but once you get into the coding, there was a substantial amount of work on this. But so of course, but now that we have permanent staff working on it, I'm kind of confident that we will be able to keep all these things updated. Yeah.
Mm-hmm. Great, thank you.
Okay, great. Thank you very much, Jean-Loup. Due to time, I think we're going to go to the next speakers. Thank you very much, Jean-Loup.
Thank you.
And so the next speaker is going to be Professor Rainer Breitling. Jean-Loup, if you could stop sharing, then maybe
yes,
Rainer can start sharing.
I have to find out how to do this. I'm sorry.
So if you go,
yeah. Okay, got it.
Yes. Yeah. Perfect. Thank you. So Rainer, if you could share your slides, start sharing slides. Yes. So Rainer is a professor of systems biology at the University of Manchester and Manchester Institute of Biotechnology. And he's also a PI in the TOPCAPI consortium. And he is interested in using computational approaches to understand complex biological systems, ranging from microbes to man. And today, Rainer is going to be talking to you about computational approaches for synthetic biology, the TOPCAPI contribution. Thank you, Rainer, if you can take it away.
Thank you very much. So my talk will be a whistle-stop tour of the computational aspects of the TOPCAPI project, or at least the selection of those computational methods that we have developed and applied within TOPCAPI. It will be, let me see if I can, yeah. The start of our logic in computational synthetic biology is that we require building blocks for our systems. Specifically for our natural product, synthetic biology, the building blocks are enzymes for our biosynthetic pathways. In the first computational component, we were looking for larger building blocks, groups of enzymes that we could use in a kind of plug and play approach as ready-made ensembles for assembling pathways to build novel chemical compounds. To do that, we relied on a collection of annotated microbial genomes, about 1,000 or so species. At that point, we identified ortholog enzymes in these biosynthetic gene clusters that we find in microbial genomes regularly. And then we analyzed them statistically, trying to find enzymes, pairs of enzymes that co-occur significantly in those biosynthetic gene clusters. They are either co-occurring by being neighbors within gene clusters, or they are just present in the same clusters. Using the significant interactions, the significant co-occurrences, we built a large network of all the putative enzymes, and in that network identified cliques of enzymes So groups of enzymes that all pairwise co-occur surprisingly often. When we map those co-occurring gene modules, enzyme modules, onto biosynthetic gene clusters, we see that these co-occurring groups, the modules, are found in very different genetic contexts, very different overall gene clusters. They are also occurring in very different arrangements. So here's a orange, green, and blue group are 3 different modules. They are sometimes found together, sometimes they are found separate. The individual genes are arranged in very different configurations. When we then, for those clusters where we know the end compound, and this is a selection where this is the case, look for the corresponding chemical structures, we realize that each of these statistically defined modules and completely unbiased identified gene modules corresponds to a chemical moiety in the end product. So the orange gene cluster corresponds to the enzymes that are responsible for the aminocoumarine biosynthesis, the blue module for deoxysugar biosynthesis. They co-occur because they are carrying out a shared function. So that indicates that it should be possible to transfer these modules as a functional unit from one cluster to the next, including to a novel, fully engineered gene cluster, and it should be able to carry out its function in an integrated fashion. A second computational step was again quite obvious to do in a project that is focusing on the overproduction of metabolites, natural products. And that's the modeling, predictive modeling of metabolism using constraint-based genome scale models. We started by producing an updated version of the model for one of the model actinomycetes, Streptomyces silicola. We had built a model for that about 10 years ago. It had been updated by another group, and within TOPCAPI, we completely recurated using the entire biochemical literature again, and we dramatically increased the performance, in this case, the prediction of the dynamic growth curve of the strain in the newest updated model is massively improved. It's much closer to the experimentally observed growth curves than in the 2 precursor models. The reason for that is, on the one hand, that we added additional biosynthetic pathways, but mostly that we recurated oxidative phosphorylation and the biomass composition in this species. So we get this global improved performance, but we also get a dramatically improved performance at the more detailed single flux level. So the top 2 figures show the correlation between predicted fluxes in the model and the observed gene expression of metabolic genes, so genes encoding for enzymes. And one can see that while there is a considerable fraction of genes that correlate with the predicted flux of the associated reaction, that's the green peak to the right, there's also a large number of genes that are actually poorly correlating, not correlating at all, or even anti-correlating with the predicted flux. In the updated model on the bottom left, one can see much improved performance. One can see this green peak is much higher and sharper. One has lost this cluster of uncorrelating genes. And if we focus on those genes that are actually showing expression changes over the growth curve, almost all of them, almost entirely, they show a good correlation with the predicted fluxes. There's still a small group here of genes that are anti-correlated, and those require individual interpretation. They tell us something about biology that we had missed still when we built the model, especially regulatory phenomena that are rarely fully represented in a constraint-based computational model. Now, this model is now good enough that we could use it as a template for building additional metabolic models for the actual target chassis that we are developing in TOPCAPI for interpreting the transcriptome data, the metabolome data that we collect in TOPCAPI, and to inform the actual engineering activities by the TOPCAPI partners. And those applications, they are currently in preparation for publication. Now, applying constraint-based metabolic modeling is a standard approach within metabolic engineering. It's very easy to do because one only needs the genome annotation. One doesn't require any kinetic parameter information about the enzymes that are involved in the reactions. But we also wanted to push the envelope a bit, and we wanted to apply kinetic modeling within a synthetic biology context. We had to accept that the data will be incomplete, that the parameter data will be noisy for those highly non-model organisms that we are targeting. We are not dealing with E. coli or yeast in these projects, so we knew that we wouldn't have a large amount of kinetic data available. So we had to develop a framework, a pipeline for building kinetic models that capture the uncertainty of the parameter values and can represent that. So we developed a protocol that allows you to translate your uncertainties about parameter values in a distribution of plausible values for parameters that we can then use as a basis for an ensemble modeling approach that allows predictions that are associated with confidence intervals and that fully represent which parts of our predictions are independent of the actual specific parameter values and which of them are highly volatile in relation to parameter choices. Those are the ones where we perhaps need to do targeted experimental work. Now, once we had developed the protocol for doing that within TOPCAPI, we applied it specifically to modeling signaling circuits, the gamma-butyrolactone system of Streptomyces silicola. It's one of the upstream regulators of antibiotic production, and we hope to apply that as a regulatory device for engineered pathways. The picture here shows the current understanding of how this circuit works. I'm not going to go into the details here. It's a relatively small system, perhaps a dozen or so kinetic parameters, but there is quite some uncertainty still about how it actually works. And we tried to test different hypotheses about the function of this system, despite a limitation in the actual experimental data available. So we applied the ensemble modeling approach. We ran through this pipeline that we developed. The result was that we basically don't know very much about this pathway on this circuit. So the function of it is still uncertain. There's still quite large areas where we don't fully understand the mechanism. But with this ensemble modeling approach, we could reject a number of previous suggested mechanisms that are no longer plausible, no matter what the specific parameter values might be. We also learn, and that is shown in this figure, where our initial belief about parameter values that are plausible might be in need of an update once one compares the predictions to the experimental data. So in these pictures, it shows the results for 4 parameters in our system that are affecting the function and dynamics of the regulatory circuit. The purple bars, the positive values, show parameter values that are becoming more plausible once we have compared predictions to models. And the purple bars that go to the negative direction, those are values that are no longer considered very plausible once we compare the predictions and the experimental data. The blue line, so these bell curves, are the original parameter distributions that we considered as plausible when we started the model. And you can see here quite nicely, this is a log scale for parameter values. These are very broad distributions. So we don't know much about the plausible range of these parameters when we start. But with our ensemble modeling, we can refine that considerably. Now, until now, I have presented methods that are really contributing to the topic of today's session, the design of biosystems. In TOPCAPI, we also develop computational methods for the other stages of the design, build, test, learn cycle. Here I'm going to show briefly some of the work we did for the test phase of the system. We are working on producing metabolites, so we are collecting quite a bit of metabolome data to characterize the function of our engineered microbes. For that, we developed a Bayesian approach to automatically annotate complex metabolome data. And we made use of the fact that we are dealing here with biological samples in which the observed metabolites will be related to each other. They will be related either on the basis of technical artifacts. They may form adducts in the mass spectrometer. They might be isotopes of each other. So those are metabolites that are connected to each other, but they also may have biochemical connections. Ultimately, all the metabolites in a biological system will be derived from each other in the broadest sense. So they should be connected. That means the identification of one metabolite should inform the identification of all the other metabolites, all the other signals in our metabolome data sets. A small example for that, if we initially have a mass peak for which 4 identifications are more or less equally plausible based on the mass accuracy alone, and then we find that one of them has also a potential relationship to something that could be an additional adduct of the same compound, and there's also a peak observed that could be a potential isotope of the same peak, and the other 3 identifications have no relationship to anything else in the same sample, then our posterior belief in the identification that is highlighted here in red increases dramatically, while the others become almost impossible, completely implausible as an interpretation. In a more complex case here, these 3 identification of a specific metabolite are all more or less equally likely, perhaps with a slight preference for the last one on the right here with the best mass accuracy. Once we look at the remaining signals in the same data set and the initial probability of those other peaks in turn, we get a strong preference for those 2 identifications, a slight preference for one of them, but that's probably just noise. We know that because this is a simulated data set, both of these are actually present in our simulations, while the initial favorite is clearly discounted because it doesn't link to anything else biologically or technically in the same sample. And that is not plausible. That means this one is downranked. Now, this approach has a big advantage that it can learn. One can change our initial beliefs from being informed only by the mass accuracy to being informed by our previous experience. What kind of other signals have we seen in similar samples before? That changes our prior expectations. It also changes our belief in certain relationships between compounds. If we know that a certain metabolite is always observed with 2 adducts of a specific type, we are going to look for both of these, and we have to find them to believe in the identification, just like a manual annotation would work. Here we can automate that. We get statistical evaluations of our confidence in the identifications. These are no longer arbitrary. These are based on the statistical calculations. We also don't discard any identifications. So we rank them, we assign p-values to them, but we are never saying this is absolutely impossible. We leave it for the experimentalists to, in the end, make up their mind based on this guidance that we provide here. The same statistical concepts based on Bayesian statistics we also use for the build stage, the computational tools there. For example, for the verification of engineered strains, we are using a high throughput sequencing approach. We have developed a barcoding strategy based on PCR, where the barcodes are encoding the columns and rows of a multi-well plate. We then pool the samples and we perform nanopore sequencing of the entire pool. We deconvolute it and we use that to verify the sequence of our constructs. Now, computationally, the challenge in this is presented in this figure. On the one hand, nanopore reads tend to be noisy. There is quite some variation in the individual reads. They are very long. That is extremely useful. They're cheap and fast to generate, but they have this noise. Now, if there were only noise in the samples, we could sequence deep enough, do a pile up, and we get a consensus sequence. We can make up our mind what the sequence information would be. But there is an additional challenge, and that is that there is a strand bias. We get different sequences in some cases, depending on whether we are sequencing the forward or the reverse strand. And the top row here shows an example of that. We, in the forward strand, get lots of A's in this position. In the reverse strand of the same sequence, we get G's. Now, interpretation of that is that there's technical artifacts introduced by the nanopore sequencing equipment. Computational, we integrate this information. We try to make our call of a mutation only if the information on both strands is consistent. We try to take into account the expected error rate and the expected rate of mutation. And we again calculate a p-value for our individual base calls at each position. We don't decide just on a single consensus sequence at each position, but we report all 4 nucleotide options with associated p-values, and users can decide on what kind of threshold they are willing to accept before they send on a construct or a strain to further characterization in the experimental pipeline. Okay, so I've walked you through the entire design, build, test pipeline here. Of course, all this work required the help of all the other TOPCAPI partners. Some of them also working in the area of computational biology, and they have developed a number of additional tools that are contributing to our work. Also within my group, the 3 TOPCAPI postdocs are supported and work very closely with the remainder of the group that are contributing their ideas as well. And I also want to thank our collaborators within the SynBioChem team at the University of Manchester, as well as the team of Professor Fukusaki at Osaka University, and the Shiki Factory team, another H2020 project that also contributes ideas to this project. And I thank you for your attention. Thank you.
Thank you. Thank you very much, Rainer. I'm looking to see if there's some questions. There's no questions at the moment. Please write in your questions, but I can start perhaps, Rainer. So you gave us a very nice overview of different softwares that can be used on one part in the design, but also in the analysis of metabolic untargeted metabolite analysis, let's say, to user softwares. They're quite different. The softwares have quite different aims, I think, each of them that you've just described. What do you think is the most, there's 2 things, I think. What softwares are most difficult to develop? And on the other hand, what are the fun softwares to develop?
That's a really, really challenging question. I think some of the statistical methods obviously are challenging to develop because they require a lot of technical understanding to do this. The ensemble modeling to do that in such a way that you capture, for instance, the thermodynamic dependency of parameter values. That's really, really tricky, and one can spend ages doing that. But on the other hand, that's also fun if one is on top of it. So if one knows what one is doing. I think the there are multiple challenges, and all of them come down to the interaction between the computational work and the biological work. If one has a clear understanding of the biological question that one is trying to address, it becomes easier to develop a method that is pragmatic, that is solving an actual biological problem. And that then also turns out to be fun, because the real fun is coming in a project like TOPCAPI when you see the experimental partners actually going into the lab, spending weeks, and it's difficult to work with these Streptomyces, Actinomyces strains, spend weeks trying to test the ideas that come out of a computational analysis, whether it's analysis of transcript or metabolome data or a design that we build. But that's where the excitement comes in, the interaction, and that's what all of these methods are about. They were initially inspired by a collaboration with a biological group.
Okay, great. Thank you very much for this nice answer. We have a question. I will read it out for you. Are there fragmentations spectra included in the tandem mass in the automated annotation of compounds?
Brilliant question. They are not in the current implementation. They have been conceptually included since we started this, because they are an obvious extension of this method. The fragment data and also ion mobility data. Those are the two sources of information that we are most interested in adding to the tool.
Okay,
it would be extremely helpful, obviously.
Yes. Okay, thank you. Thank you very much. Due to time, I think we're going to go forward now with our next speaker. If Ivan can share his presentation.
Sure.
Yeah.
Yes.
So the next speaker is Ivan Domanzian, and he's a PhD student with Jens Nielsen and Verena Sievers at Chalmers University of Technology. And of course, he is in the team of CHASSY, and he's working on integrative multi-omics data analysis and software development, including the expanding the Gecko software. And today he's going to talk to us about learning from enzyme limitations to improve in silico strain design. Please go ahead, Ivan.
Hello, everybody. Do you see my slides?
Yes.
Yeah. All right. So let me get started. Yes. All right. So, yep, I have already been introduced. I'm Ivan Domanzian. I come from Chalmers University of Technology. And let's go through this presentation. So, metabolism, I think that we all here know that metabolism is at the very core of cellular function. It's a huge and very complex network, but it's very interconnected and also highly constrained by thermodynamics, kinetics, and even environmental parameters. For probing metabolism in a quantitative way, the community has developed the concept of genome-scale metabolic models, which are a computer model of real cells that comprise all of the biochemical mechanisms that take place inside of the cell. And well, these models can be seen as very, very comprehensive knowledge bases of living cells. We use genome-scale metabolic models together with flux balance analysis to get quantitative predictions of metabolism. The inputs for these predictions are exchange flux of nutrients and byproducts, but also biomass composition. As outputs, what we get usually is distribution of internal metabolic fluxes, meaning the reaction rates for all of the reactions of metabolism inside of a cell. We can also predict cellular growth rate by these type of models. And these models usually work on the assumptions that there is no internal accumulation of metabolites, meaning that cells work at steady state, and also that cells operate according to optimality principles. For example, the most used objective function in GEMS is the maximization of growth, assuming that cells have evolved to maximize growth with the resources that they have available. But well, a problem of GEMS that is very well known is that multiple solutions may exist for the same optimal objective value, meaning that multiple flux distributions can hold the same observable phenotypes, let's say. And this optimal or this alternate optima may arise because of several reasons. One of them being that metabolism is intrinsically redundant. So cells have evolved to be robust and to have different alternatives to achieve the same objective. But these multiple solutions may also represent our own lack of knowledge when developing these models. And this is due to the fact that typically in GEMS, kinetics and allosteric regulation is not accounted for. Just some reminder on the limitations of metabolism. So we have said that it is a very interconnected and complex network, but we humans also know some other complex networks. And a good example of this is cities. So here I'm showing a map of Mexico City. And if we zoom in one of the streets, like in the center of the city, we can see that this is a very narrow street. And because of its characteristics, this street is just capable to sustain a very low traffic of cars at a given amount of time. In contrast, if we zoom in to another street, we can see that these events can be taking place at the same time, but it's just that one street is very narrow and can just take a few cars, and the other street is very wide and can take a very, very high traffic of cars or flux of cars. So this suggests the conclusion that not all streets have the same limitations. And if we go back to metabolism, not all pathways have the same limitations. And why? This is because enzymes, each metabolic reaction is limited by its corresponding enzyme. So enzymes are very specific, and enzymes have evolved to have very different catalytic activities. Therefore, pathways will have different limitations, and we would like to model this or to incorporate this into our models. We can do this because there is a very well-known relationship in biochemistry, and we know by heart that metabolic fluxes or reaction fluxes cannot be higher than the product of their kinetic constants or their kinetic capacities, the kcat values, multiplied by the abundance of the specific enzymes that catalyze reactions. And considering this principle, we developed GECKO in 2017, which stands for Enhancing Genome Scale Metabolic Models with Enzyme Constraints Using Kinetics and Omics Data. And what we do with GECKO is that we take a pre-existing genome scale metabolic model and we incorporate all of the enzymes that we are able to find information for in public databases. We incorporate them as part of the network. All of these enzymes constrain the fluxes that the different reactions can take by the equation or the inequality that I just showed. And all of the kcats that we feed into these models, they come from publicly available databases, mainly from Brenda. And the Brenda database is an amazing resource. We have entries for thousands of enzymes. And in GECKO, we have a pool of 60,000 kinetic parameters that can be used to constrain enzyme constraint models of metabolism. Additionally, if we have condition-specific proteomics data, we can use this proteomics to further constrain the solution space of the models. And at the end, what we get is a so-called enzyme constraint model that I will refer as EASY model from now on. The first implementation of the GECKO toolbox and the GECKO formalism was applied on a model of Saccharomyces cerevisiae metabolism. And we proved that this formalism was able to improve majorly the phenotype predictions for not just for the wild type cells, but also for mutant strains. So this model was able to predict the Crabtree effect, the switch from respiration to fermentation, even in the presence of oxygen for Saccharomyces cerevisiae. But we were also to predict with very high we were also able to predict cellular growth on very diverse environments with this model, with a very minimal amount of experimental data used as constraints. So this all looked well for cerevisiae. And well, we use genome scale metabolic models and flux balance analysis. All of these are part of the family of constraint-based methods. And as you can see here in this slide, there are plenty of constraint-based methods that have been developed for specific biological questions. But there is a specific family that I would like to focus on, which is the constraint-based methods for strain design, and in particular, the method of FSCOF. In brief, FSCOF is a method in which we take a genome scale metabolic model and base it on the assumption, or we know that there is usually compromise between maximizing cell growth or biomass formation and maximizing product formation. So they are usually compromised. What we do with FSUF is that we perform a series of simulations spanning both objectives, going from maximum biomass towards maximum production. And in doing so, we identify all of the reaction steps that are consistently increased in their flux for enhancing production, and all of those that are consistently decreased for enhancing production. And this might suggest, when we map these reactions to their encoding genes, this might suggest gene candidates for overexpression, knockdown, or even knockout. This is a very highly used method. It's a very cited paper, and it might be highly used because it's conceptually very simple, it's very easy to implement, and it's readily available in the most used softwares for constraint-based methods. However, there are some problems, and namely, FSUF predicts a large number of candidates for a given product, usually between 50 and 100 gene candidates that need to be tested. And a lot of these genes might be false positives, and they may arise because of the lack of kinetic and regulatory information for enzymes that we have in genome scale metabolic models. And a lot of these targets that are suggested might be even redundant targets. So the natural question here is, what is the optimal combination of gene targets for enhancing production once that we have predicted them? So what we did is that we put together this pre-existing method, and we made our own implementation of FSUF and incorporated it into the Gecko toolbox so that we can use it with the enzyme constraint models that are generated by it. So we run the method for any product of interest. We generate list of candidates, and it's the same. We get tons of targets to test, but we take advantage of the features of easy models. And as we have all of the kinetic parameters and the information for the enzymes that are acting in metabolism, we use this kinetic information in order to assess, for example, like if there is redundancy in the targets that are suggested, or these kinetic parameters can also help us to prioritize and assign different levels of priority to our targets. Oh, sorry, something happened with my slides. Sorry, could you see my slides again?
Yes, it's fine, yeah.
Sorry for that. This allows us to filter or to narrow down the amount of candidates. We also use easy models because we have the enzymes in an explicit way in our models. So this allows us to simulate mechanistic genetic modification. So we can either knock out the enzymes in our model, or we can overexpress them by forcing an increased usage of this enzyme. And this is allowed by easy models and not common in traditional GEMs. At the end, what we do is that we follow a combinatorial approach, and taking all of this information in which we rank the gene candidates and we prioritize some of them, we combine the top targets and assess that the final strains are feasible strains and productive strains. So this allows us to get optimal combinations of targets for our strains. We have called this method robust enzyme constraint FSUF. And for validation, what we did is that we run this fully model-driven approach, which is we wanted to produce heme with Saccharomyces cerevisiae cells. So we ran our method, and initially FSUF suggested around 100 targets for engineering. But when applying all of these levels, all of these multi-step, all of these different steps in the method, we narrowed down the list of targets to just 11 genes. We went to the lab, modified these 11 genes, encompassing deletions and overexpressions. And just with engineering these 11 genes, we were able to get a 70-fold increase in intracellular heme concentrations, so yielding a highly productive strain. So here what you can see is that it was obvious that our cells were producing more heme, because we can see the red color of our samples. So we were very happy with this. We validated our method a bit further. So this is another example from our lab in which, following a purely rational engineering approach, our lab developed this strain for enhanced spermidine production with Saccharomyces cerevisiae. So we run the same, but with our method, just to see if there was any overlap between a purely rational engineering approach in comparison to a model-driven one. And we saw that there was a very high overlap of the genes that were tested and the predictions that were suggested by our method. Moreover, we also saw that there are some other targets that are also predicted by our model, but were not tested by the experimentalist people, suggesting that the method could find other targets that are usually ignored by purely rational engineering approaches. And this is because, well, metabolism is huge and complex. Also, what we show with this is that the method is able to suggest complex strategies that involve the coordination of different pathways in order to increase production, as in contrast to just suggesting targets in a linear pathway. In CHASSY, we are very occupied with producing aromatic compounds. And one of those compounds has been 2-phenylethanol, which is known because of its rose aroma. So at TU Delft, our collaborators, they developed this strain, also following a purely rational engineering approach. And here you can see all of the modifications that they incorporated into this strain. So we ran our method as a validation to see how many of these targets we were able to capture. We're very happy to see that almost the full metabolic engineering strategy that was followed by our collaborators was also predicted by our method. So here we have to say that this strain was yielding a titer of 1.6 grams per liter of phenylethanol, which is remarkable because we went from nothing to these titers. So this was another case of success, and this provided us with confidence for our method. So we went further, and together with our collaborators in France, we also applied this formalism to a Yarrovia lipolytica strain for the same product. So we wanted to enhance the production of 2-phenylethanol with Yarrovia lipolytica as a strain. And what we saw is that once again, here we followed a mixed approach. There was an initial suggested of targets, and as here you can see highlighted those targets that were predicted by the method. But the strategy was also complemented by rational engineering targets that were suggested by previous knowledge. So this strain reached a comparable titer of 1 gram per liter of phenylethanol. And we even went further in the CHASSY project and applied the same approach. So it's a mixed approach between the model suggestions, which you can see highlighted here, the targets that were suggested by the model. And this is using Cloveromyces marcianus as a CHASSY strain, but also our collaborators in Cork, in John Morris's group, they also came up with other targets suggested from their previous knowledge. So when combining these targets, we achieved these titers of phenylethanol production using Cloveromyces marcianus. So here, what I have shown is that our method works for multiple products. Our method is able to provide us with predictions that encompass the coordination of complex metabolic engineering strategies, and that it also works for several organisms, meaning that it works for several models. So we want to take this concept further, and now we are exploring the production of 120 different chemicals with Saccharomyces cerevisiae. So here you can see the kinds of chemicals that we want to produce. We have run simulations or predictions for all of them with our method. And here, just briefly, what I want to show is that is the drastic decrease in the number of targets to test that we obtained by our multi-step method. So FSCOF tends to suggest a lot of targets, as I have mentioned before. So with our multi-step method, what we get is a very reduced list of candidates predicted to be an optimal combination that can be tested in the lab. Just to mention, we have the enzyme constraint models for these 3 industrially relevant yeast species, Cerevisia, Lipolytica, and Marcianus. But recently, we have developed enzyme constraint models for other model organisms, such as E. coli and human metabolism. We have put out a preprint. So here you have the link to our preprint. So we have all of these models waiting to be used by the community. All of our tools are open for community development. So here you have the links to the GitHub repositories in which we have stored all of our software, but also all of our tools. And briefly, I just want to mention that we have recently partnered with DTU in Denmark and the Center for Biosustainability. And together with them, we are plugging in our enzyme constraint models into the caffeine platform in order to allow or to enable web-based simulations and web-based design of cell factories. So here you have the link to the caffeine platform. It's a very user-friendly tool. and you are very welcome to use it and collaborate to its further development. So as a summary, I would like to say that GEMs are extensive knowledge bases of metabolism, but their predictive power for metabolic engineering is still limited. If we incorporate enzyme constraints into GEM, we can improve the phenotype predictions and even provide insight on protein allocation and kinetic bottlenecks. Our method that we have developed, Robust Easy FSCOF, leverages the simplicity of the FSCOF algorithm and the features of enzyme constraint models. Robust Easy FSCOF predicts complex metabolic engineering strategies for design of optimal production strains that can work together in vivo. The method can be applied on any functional EC model for any organism for prediction of gene targets for native, but even for heterologous products. We have produced a catalog of high quality EC models that is already available for you to be used. And all of our models and software tools are free and open to community development. And I would like to thank all of our collaborators that are also part of the CHASSY project. So you can see that this involves a lot of institutions, and that's all.
Okay, thank you. Thank you very much, Ivan, for a very nice talk. I will look for the questions. Here's one question. What's the difference on how well FSCOF works on EC model versus conventional GEM?
Yes. In principle, they are the same. So FSCOF is the same. So the main addition is that we added these further steps of validation of the targets. So it's not that we just keep the targets that are suggested by FSCOF. We use the information that is in EC models in order to rank our targets based on kinetic parameters, but also the information in the model can allow us to find which targets are redundant, and we discard those redundancies as well. Yeah. So it's like, it's more, this method, Robust FSCOF, is more about the different steps of validation that we have. And at the end, we shouldn't forget that the EC model also allows us to find the optimal combination of these targets and to get a quantitative assessment of these strains.
Okay, thank you. Thanks. Rainer, do you have a question, perhaps? Because we can't.
Yeah, actually, I would have a question. So you described the gecko approach where you constrain the constraint-based models based on experimental data using kinetic parameters from render. Now, the obvious question for me would be, can you combine that kind of approach with ensemble modeling? Can you somehow make use of the uncertainty, somehow quantifying that these parameters will not be exact for your specific condition? They will always be associated with uncertainty and lack of confidence.
Exactly. This is a very interesting question, and I would say natural question when we talk about kinetics. Probably not in a straightforward way, but I can see that we can produce population of or families of models taking into account uncertainty of parameters. However, the combinatorial space will be infinite and will be untractable very soon. But something that we do in Gecko is that this is still constraint based. So it's not unique solutions. So what we want the enzyme parameters is to establish or to impose upper bounds on the fluxes. So the Gecko approach is quite conservative in which k-cuts to pick. So we go whenever we have multiple parameters reported for the triplet of organism, EC number, and substrate, we go for the higher ones that are reported, for instance. And we also have, when data is missing, we have incorporated a phylogenetic distance-based algorithm in Gecko in order to choose a better match for kinetic parameters. So this has allowed us to generate enzyme constraint models even for poorly studied organisms. But yeah, so this is what short answer is like, we take a conservative approach at choosing kinetic parameters. And as we just use them as constraints, we kind of avoid the need of generating or taking all of this uncertainty into account.
Yeah.
Okay, thank you. I have another question here.
Yes.
How easy are your tools to use for a novice?
Okay, so easy models are compatible with the established tools that we have. They are compatible with Cobra, with Cobra Pi, and with the Raven toolbox. We have generated this pipeline of easy models containers. So all of these models are continuously updated every time that the original sources are changing. Our continuous integration pipeline detects and updates the models. So if the users want to use them, they are always going to get the latest version. And we are trying to move forward with this, and that's why we are now integrating it into the Caffeine platform. So in the Caffeine platform, we are starting to provide this visualization of enzyme utilization with easy models.
Okay, thank you. John, do you have?
Yeah, I have a question, Ivan. Thanks for the presentation. And I'm just thinking in terms of maybe linking a couple of things. But, you know, in the first talk, Jean-Loup talked about retrosynthesis and design of, let's say, completely new pathways. How, maybe following the last question, but how easy would it be for somebody who did that and designed a new pathway to drop it into one of your EC gems or into several of them and to try and decide to use your model and your robust FSOF to kind of come up with improved strains to produce new products like that?
Well, it's as easy as it would be to implement a heterologous pathway in any gem. So it's like, I would say it's easy for a modeler because we modelers are used to work with models. So for any person that deals already with the Cobra Pi package or with the Cobra toolbox, it's the same format. So they can just incorporate the pathways in the same way. However, this is facilitated by the Caffeine platform, and we are still working there to really ensure full functionality of the web-based service, so that what we want to do is like just offer a catalog of pathways, or even the user can upload their own pathways and they just plug into a model. So we are working towards that direction.
Yeah, if I may intervene, because this is linked. So we have a tool that's called RP-SBML in the Galaxy platform that can plug a pathway generated by the whatever retrosynthesis software into an SBML model. So
perfect.
It is just an SBML format. Okay, that's
yes. And easy models are produced both. Whenever we produce an easy model, they are produced in the MATLAB format, but also in the SBML format. So it's like probably the compatibility is there.
Since my mic is on, I had a question. Can I ask it? Oh, it's too late.
Yes, go ahead. Yes.
Yeah. So in the formulation, at some point you showed that you were adding some constraint. Okay, and it was V lower than KCAT E. Okay, and so this is assuming the enzyme is saturated. Okay, when you do this. Okay, so it's saturated by the substrate. So is it a reasonable assumption that?
It's a very good observation. Well, at the end, all of the enzymes are connected to the protein pool, and that protein pool is equal to the total amount of enzymes mass that are present in cells. So this is an experimental parameter. And what we do is that we assume an average saturation of all of the enzymes in that pool. So basically, we just multiply that enzyme mass times the saturation that we want. And this usually for easy models at high growth rates, we assume an average saturation of 0.5. So that saturation is applied to the whole pool, but not to the individual enzymes.
Okay, got it. Okay, got it. Okay, thanks.
Okay, great. Thank you. I have one more question from the audience. Can this model be used to improve flux of heterologous enzymes and pathways? So if you're introducing something synthetic, basically.
Yes. So far, our experience has been more in like whenever we introduce something synthetic, this method enables us to find the bottlenecks in the native metabolic pathway. So it might be the case that your pathway, your heterologous one, is already very efficient, but there might still be some bottlenecks inside of metabolism. And this method is suitable for that, for finding the native bottlenecks. So what do we need to rewire in metabolism? Yeah. But I would say that it can help us to understand, once that we have incorporated a pathway, how can we take our strengths even further? That's it.
Right. Okay, great. Thank you. I have one quick question, if I may. You show that you have your model now for 1, 2, 3, 4, 5 different organisms, from E. coli to humans and yeast. Are you thinking of actually also expanding this to other microbes as well? There are quite a few different groups working on different microbes for different purposes. Like, for example, for our TOPCAPI, our host is Acknowledgements or Streptomyces, just because we're trying to work with antibiotics. And sometimes we find that it's very difficult to use our strain because no one wants to really work on it very much. So I was just wondering how easy is it with your EC models to expand it into different organisms?
Well, I should say that that's probably the main topic of my PhD. So I have worked in order to generalize and facilitate the usage of this methodology. So let's say when I just started to generate the first alternative EC models, it took me a couple of years, but now it's a more plug and play tool. So now we provide this tooling, which is very, very easy to generate for new models. And we are running tests in CHASSY. So what we are doing in CHASSY is that for all of the gems that we have for any organism, being microbes or even multicellular organisms, we are incorporating them into this pipeline of EC models containers. So now it's much more user-friendly.
Yeah. So how many months? Oh, sorry. How long do you think it takes to actually get this up and running from scratch?
A day, 1 day, let's say. Of course,
1 day.
Yeah. 1 day once the one has collected the data that is necessary to run the tool. So yeah.
Okay.
Yeah.
Great.
And
Thank you.
the process of enhancing a model with enzyme constraints in the case of Cerevisiae, it takes around 1 hour of computation.
Okay, great. Yeah. Thank you very much. Okay, so thank you. I'd like to thank all the 3 speakers for a very nice presentation today. And I have taken some time for general discussions, and if anyone has any questions, please do let me
Rico,
know.
I had one more question. This is John
yes,
again.
Please, go ahead.
This was actually a question for Rainer, because earlier in your talk, Rainer, you mentioned what I thought was really nice, where you found these, I'll call them co-occurring genes for making particular chemical entities. In the project, did you do any experimental work to follow up on that, where you essentially took several of these sets of genes and just tried to make, I don't know, a completely new molecule or something?
No, we didn't. We relied on historical tests for this. So we compared our modules to what people had already described for biosynthetic pathways, for those where the compound is known and where the biosynthesis was known. We haven't yet taken it to the lab ourselves to actually test how far can they be plugged together, and how easy is that, and how distant can the target pathway be. So that's all things that need to be tested, but it's a starting point.
Yeah.
There are thousands of these modules that are very, very highly statistically supported and come from very different contexts. So they are apparently in evolution deployed very, very widely.
Okay, because I think it's fascinating, and one of the things I was wondering, was whether there would be any, I don't know, constraints on, I call them the linkers, that, I mean, it's one thing to have sets of components, but to actually, how do they get joined together type of thing?
Exactly, and it's probably similar to what you see in the module, say, for modular polyketide synthase, where these interactions happen within an assembly line. There must be some constraints on the handover between the different modules.
Okay. Before we go into the general questions, I have some general questions for all 3 of you. Perhaps you can answer. But before we go there, there's one question from the attendees saying that, where can we find the recording to the webinars? Can you provide us with the link via email? We can't send you an email, but it's very simple. The webinars will be uploaded into either the CHASSY or the TOPCAPI websites. But the actual time frame when this will be done is, I am not sure. I am kind of looking into Verena or Rebecca to answer when this will be uploaded?
I can step in there, Erico. I'll aim to upload them tomorrow on the CHASSY website.
Okay, and can you do that on the TOPCAPI website too?
Sure, they'll be available on both.
Okay, great. So tomorrow in both TOPCAPI and CHASSY websites, you'll be able to find this webinar. So I know we're almost time to close, but I have one question for all three of you, because you gave us a really nice overview of all these different softwares from design to analysis. How practical do you think all these softwares are for industry to use them? Coming from an industrial point of view. I don't know who wants to go first. Whoever wants to go first.
Maybe myself. Yeah, we do have industry using the Galaxy platform. So I guess they find it easy enough, easy enough to use.
Okay, so you're saying that there's quite a few industry companies using your Galaxy?
That many, but we do have industry.
Some. Yeah, okay. Yvonne, maybe you were saying you want to say something?
Yeah, for us, it has been a kind of a long story. So it's like we have had some industrial partners using Gecko, but they have used it for the well-established cases, so working a lot with Cerevisia. So that's why we want to move forward and integrate this into CAFE to really bring it to a broader or a wider audience. Easy Models container, like this continuous integration pipeline, is also a step forward in that direction, in the way that we don't want the users to take, basically to spend any time on updating their models, so that they can always just pick the most updated tool. So that is already implemented, that is already provided, let's say. But there's a key thing here, which I think that is like, well, these models, for instance, constraint-based methods or models, they depend a lot on the constraints. So it's like, even if we work a lot on the interfaces in providing the industrial partners with very easy to use tools, they still require biological relevance of the constraints and the data that are used as inputs. So that is always that we must never forget.
Yeah. Yes, I understand. Yeah, thank you. Rainer, do you have something comment?
I can't add much more there. I think the industrial researchers will face the same limitations as a biologist who would try to do this on their own. It's usually relatively small teams. And for that, you need an established tool. You need something that's already gone through several rounds of improvement. Both Ivan and Jean-Loup have presented the challenges of getting from a pioneering idea to something that anybody can use robustly and can install and doesn't have to worry. And even then, as Ivan rightly says, you still need to understand the biology. You need to know what you are doing, because any bioinformatics tool, any systems biology tool can give you numbers, can give you p-values that look great, but if you don't know what they mean and what's happening there, it can also very quickly lead you in the wrong direction. So it will remain an interesting challenge, both for industry and for biologists who try to use this without a close collaboration with the systems biology group.
Right, great, thank you. John, do you have a general? Oh yeah, Ivan, go ahead.
Last contribution, I would say that in the same line. So we researchers or we developers, yeah, we should do a lot in developing user-friendly tools, but we must not forget about the communications, which is, let's develop good tutorials, let's write good papers that are well explained, and let's write documentation for our software. So that is always necessary, and the user base is going to depend on that at the end.
Yeah, great, thank you. I have another question from the attendee. It says, for Ivan, how much data do you think is enough to produce reliable results for using your tool? How much data do you need, I guess? Or how much data do you think is enough to have to produce reliable results using your tool?
At the end, the generation of an easy model depends on how well represented the target organism is in the Brenda database. So we can make sure that for the well-studied organisms, the model is going to be high quality. For the poorly studied organisms, in Brenda, the model is probably not going to be that high quality, but we have parameter curation scripts that can aid into improving the quality of the model. So it's like initially the user doesn't need to provide any data because this data is going to be retrieved from the public databases by the toolbox itself. So one can start just with a gem. That is the initial part that we need, a gem. And if you have already a gem for your organism, it's a matter of trying to plug it in with Gecko.
Okay, great. Thank you very much for the answer. John, do you have any general questions that you'd like to ask all the three of our speakers today?
I suppose my kind of general observation stroke question is that, in the literature, there is a wealth of microbial cell factories described for producing many very interesting molecules. And very few of those have ever been applied commercially or in industry. And my question is to what extent do you think the kind of the approaches that you talked about are the game changer to kind of to really make this step change that we can make cell factories a real reality? Or are there other major bottlenecks as well? I mean, are ye the solution to the world problems?
No. I'm speaking for myself. No, there are other issues. I mean, when you scale up, the Galaxy, what I presented, the Galaxy platform, it is not for scale up. Okay, it is not a tool that is dealing with that is dealing with what's happening in the bioreactor. Okay, so there are other issues, heterogeneity in populations, and there are many issues that are in bioreactors that are not yet tackled by the tool that I presented. Okay, so so far what we have is something that is upstream. Okay, so upstream of that, making sure you produce the compound in sufficient quantity so that you can start wondering if it worth going industrial. Okay, but we are not tackling that problem. There are tools out there that exist now, but maybe not as much as the classical metabolic engineering tools. But there are ways. Okay, also, for instance, design of experiment. It is something that is also being used in bioreactors to optimize the process. But that does not take care about the heterogeneity. I think this is one of the main issues. Mm-hmm. Mutations, and so.
Okay. If Ivan and Rainer don't have anything else to add, then yes, Ivan?
I think that, just briefly, I'm a chemical engineer by training originally, and then I moved to biology. And I spent some time modeling reactors. And something that I have observed, according to my experiences, when one is modeling reactors with very specific formalisms, focusing a lot on the hydrodynamics and all of this, one usually oversimplifies the biology of the bioreactor. So, oh, this is just a monod kinetic expression, whatever. And the same on the other side in biology, like, oh, we can have very sophisticated models of the cell. And then when we want to model the process, we oversimplify the processes and don't take into account a lot of things. So the challenge, I would say, that is in really multi-scale modeling, but not multi-scale, not necessarily multi-scale biological model, but multi-scale process modeling, in which we incorporate the best models that we have for biology, like these sophisticated, for example, constraint-based methods, together with sophisticated models of process development. And I think that the challenges are there for modelers and engineers, and that's probably the next direction in which we will need to focus.
Yeah. Okay. Thank you. I have another question from the attendee for Ivan. Could you use these models retroactively? So not to try to modify the most popular organism to produce your compound, but to identify the species and strains with the highest potential for production of your compound of interest?
That is a very good question. Like, I don't know, like just quickly what I can think about that will rely on. I cannot think of a straightforward answer, but what I can think now is that we are producing a catalog of models. So basically what we can do is to try to implement something with all of the models that we have available, and this is increasing. So at some point it's going to be a lot of microbial species that we are aiming to have there, and to kind of do a quantitative assessment. So we run the same for all of them, and we just compare the predictions, and then we see which of them is the best candidate.
Okay.
But there might be probably better approaches to this.
Yeah. Okay, Rainer, you had some.
I just wanted to add, we have done that a few years ago, of course, with much less sophisticated models than what Ivan is doing now, but we tried different species. We had automated models for, I think, 35 different Actinomyces strains, and then we plugged in, I think, about a dozen or so different compound classes and tested the predicted production ability for each of them, with taking into account the trade-off between compound of interest and biomass. And it was really interesting. Of course, it would be interesting now to run that again with these much better models, perhaps a broader range of compounds. But the differences that we saw between these quite closely related species were dramatic. There were some that seemed to be universal super producers, and others that are very highly focused or fail entirely. So, and that wasn't easily predictable. So it wasn't the very complex native producers that have lots of secondary metabolites that would be ready to produce everything, and the minimized genomes would be failures. It wasn't that intuitive. So one could really learn from the modeling there.
Yeah, great. Thank you. Thank you very much. I think we could, there's no further questions. I think we should try and close this session now. I just want to share with you the last slide as advert for our next webinars. So this was really only the first start of our webinars. We have 2 other webinars which is coming up. One on the 25th of March, which is going to be on the topic of the build side within the design, build, test, learn. And on the 8th of April, it will be the topic on the validation and the scale up. And please do register for them. You need to register for each webinar separately. Just because you did it today doesn't mean that you'll be able to get into the 2nd or 3rd webinar. So please do go and register. It is all free, and you can see on the bottom here, the bottom here, here's where you can find all the registration details. So thank you all very much. First of all, thank you for the 3 speakers for very nice examples of all the design and softwares. And thank you all very much for staying on until quite a long time. Thank you very much, and hope to see you on the 25th of March as well. Okay, so thank you again.
Bye-bye.
Bye now.
Bye-bye, everyone. Thank you very much. Great
Bye-bye.
session. Well done.